let's talk about muscle. So muscle, favorite organ, love muscles, okay? Why do we need them? Well, they provide movement. They give structure to our skeletal system. Most importantly, they really participate in generating your posture, right? Your upright posture against gravity, and provides, what we already know is the #1 factor in joint stability, right? Muscles can produce heat. You know, I think about going to the gym and I already start sweating. It's just like this reflex reaction. So muscles produce heat, all right? We can communicate using our facial muscles, and we can control our body openings like our urethra and our anus, because we have muscle tissue that surrounds these orifices, okay? Okay, so what's so unique about muscle is that muscles can get excited, so they can respond to electrical impulses. Okay, and in fact, we need electrical impulses in order to tell muscles to contract, right? So without electricity, muscles will just stay dormant and you wouldn't move. Okay, so if you think about someone that's paralyzed, they're paralyzed because more often than not, they have nerve damage. And nerves give life to muscles. You don't have nerves, muscles are not going to be told what to do, and you can't move them. Okay. All right, conductivity means that the electrical activity can spread. We'll talk about that. Contractility means the muscle can shorten, but it can also relax and lengthen back into its original state, which is just as important. Okay. Muscles can stretch, so you can take them beyond a shortened position. And again, the elasticity allows a muscle to shorten or to lengthen, but to ultimately relax back into its original position, which is probably the most important thing, right? Okay, so let's talk about 3 classes of muscle, and we're just kind of summarizing here. We're going to look more at tissues under the microscope next week when we study muscles in lab, okay? So we've already talked briefly that we have 3 different kinds of muscle tissue, okay? But let's learn little characteristics about each, okay? And this will really become apparent in lab when you look at the slides under the microscope. Skeletal and cardiac muscle contain a striated appearance, and we'll learn why shortly. Striated means striped, okay? So it's like a striped appearance when you're looking at the cells. And it's because of the organization of the protein fibers, which we're going to talk about. Okay, so that's what striated means. Skeletal muscle is voluntary. What does that mean?
You control it.
So you're in control of contraction, right? You can choose whether or not to contract skeletal muscle, all right? Cardiac muscle, where do you think we find this in the body?
Heart.
The heart. Do we have control over the heart contracting? No, we don't. So that's considered involuntary, okay? Smooth muscle is unique in that it doesn't have a striped appearance. So students always confuse it with some sort of connective tissue when you look at it under the microscope. So we'll get there in lab. But smooth muscle lines the inside of your digestive organs, for example. Okay? Lines the inside of your esophagus. So these are areas, do you have voluntary control over your digestive organs? No, you really don't. Nope. So again, smooth muscle is involuntary. Okay. We'll talk more about these terms. Skeletal muscle cells are called myofibers. Okay, so again, more terminology. It's going to be a whole new language yet again. Cardiac muscle has cells that maintain the tissue, okay? Sites, this is nothing new. Cardiocytes means now we're talking about the heart, okay? And then smooth muscle, we'll notice under the microscope especially that the cells kind of take on this weird like spindle shape, meaning it comes to a point at either end, like a really pointy point, versus skeletal muscle, it narrows, but it narrows because muscles attach to bone via a tendon. So muscles will basically merge in with a tendon, which then attaches it to bone, okay? But smooth muscle has really just kind of a pointy-ended attachment, so you guys will see that in lab. So these 3 slides basically are just used for assistance with the histology portion, okay? So skeletal muscle, again, striped appearance. Here are the nuclei, and I told you before that skeletal muscle cells are one cell in the body that are multinucleated, right? So there's more than one nucleus per cell. So 1, 2, 3, a bit of 4 cells. On this cell, we can see 2 nuclei. On this cell, we can see 2 nuclei. So multinucleated cells, okay? If we take a skeletal muscle fiber or a cell and cut it transverse, okay, this is what it is. And it's just packed with protein fibers, and the nuclei of the cell are shoved to the periphery. Kind of like, what kind of tissue does this look like?
Adipose.
Adipose, right? So you got adipocytes, and they're full of lipids, so they shove the nuclei to the perimeter. Same thing with skeletal muscle cells, full of protein fibers, because the protein fibers are going to be what allows for these cells to shorten and lengthen, okay, and we'll talk more about that. Okay, so if we talk about muscle, we're gonna be using the terms myo and sarco. They both mean muscle, okay? So when you're working out at the gym, okay, and you're trying to build weight or build muscle girth, okay, you wanna get bigger, stronger, you're not developing new muscle cells. The muscle cells that you have are adding protein fibers, so they're getting bigger. So the cells that you already have are getting bigger. Okay, so that's hypertrophy. Okay, so that's hypertrophy. Those cells are not going through mitosis. You're not creating new cells. The ones that you have are getting bigger. So just as much as they can get bigger, if you stop training, they can get smaller again. Okay, so again, if you don't use it, you lose it. So your body's gonna respond to whatever kind of training or challenge that you give it, so to speak, okay? And unfortunately, the cells that make muscle fibers, the myoblasts, right, get lower as you age. So, you know, getting old is for the birds, but again, like I said before, the nice thing is that your body will respond to exercise when you're 85, just like if you're 25. You will hypertrophy your muscle cells if you put tension on them and you challenge them. And that's a lifelong dedication, right? Okay, so let's talk about muscle as an organ. Now, muscles are very highly organized, okay? Because each cell is cylindrical in nature, okay? And there are millions of cells in a particular muscle as an organ, we can group and surround different parts of a muscle with fascia, okay? And we're really gonna look at fascia when we dissect the cat. You're really gonna appreciate fascia by the time you're done with the cat, okay? So the first thing we have most superficially is what's called superficial fascia. It's also called telosubcutaneous. It's the hypodermis layer of your skin. Okay, so that's where you find adipose connective tissue. And what the superficial fascia is designed to do is it's designed to separate your skin from your underlying muscle layer. Okay, so superficial fascia is going to be more like areolar connective tissue, like we talked about. Okay, so again, that's going to keep skin and muscle in separate layers. Okay, separate compartments. Then deep to that, in between muscles as organs, you're going to have more fascia. And that's going to be considered deep fascia, okay? So this is going to be like dense connective tissue, okay? Okay. Now, as far as coverings of the muscle itself as an organ, okay, so a muscle as an organ has 3 different wrappings. Epimysium is the most superficial, perimysium in the middle, and then endomysium on the inside, okay? So, epimysium is the most superficial here, okay? It's superficial, think of it as saran wrap that's surrounding a muscle itself as an organ, and it's continuous with the tendon that attaches the muscle to the bone. And tendon is made of what kind of tissue? Dense regular connective tissue. So epimysium is a continuation of that dense regular connective tissue. Okay? All right. Now, let me just get this concept down here since we're already talking about terminologies here. Okay, if we're talking about myofibers, okay, or if we say muscle fiber, it's the same thing. Those are both synonymous with muscle cells, okay? Muscle cells that have more than one nucleus, okay, that have organelles, that have cytoplasm, okay? A myofiber or a muscle fiber is the same thing as a muscle cell, okay? All right, so paramycium is going to be surrounding collections of fibers. And a collection of muscle fibers, which is also called a muscle cell, is called a fascicle. So a fascicle is a collection of muscle fibers, and fascicles are surrounded by perimycium, okay? Which means individual muscle cells or muscle fibers have their own wrapping, and their own saran wrap is called endomycium, okay? So the way to think of it for those of you that are visual, all right, I think of like an entire muscle, okay, as like, because it's cylindrical in nature, right? I think of it as like a boba straw, right? Big boba, thick opening, wide boba straw. So it allows the boba nuggets to, you know, basically whoosh into your mouth and choke you. So you've got your boba straw, right? And then inside the boba straw, you're gonna put a couple traditional straws. So those traditional straws are going to be fascicles. And inside the traditional straws, you're gonna throw in some of those 7-Eleven coffee stirrers. Do we all know those? Do they still have those at 7-Eleven? Yeah. So you can't sip coffee out of them, but you can use them to stir. Okay, so think of those as your muscle cells, the smallest of the small. Okay? But they're all cylindrical in nature and they all fit together in an orderly fashion. Okay? Okay, that's kind of the way I like to visualize it. Okay, so let's repeat ourselves because repetition is the name of the game. So fascicles are collections of muscle fibers or muscle cells within a muscle as an organ. Okay? Muscle fibers are also called muscle cells. We can also call them myofibers. Okay? So a myofiber or a muscle fiber is the same thing as a muscle cell. Okay? Now, we can go one step further down. Inside a muscle cell, we have what are called myofibrils. Okay? You can put myo in front of most things. And myofibrils, okay, are clusters of proteins, and we can consider this the organelle level of a muscle cell. Okay, so the myofibrils are the organelle level of a muscle cell, and they're clusters of proteins because muscle cells are packed with protein. Okay, specifically two kinds of protein that we're going to talk about. And those protein fibers at the smallest level are called myofilaments. The two myofilaments we're going to talk about today are actin and myosin. Okay, so those are protein fibers, also called myofilaments. Okay. All right, lots of new terminology already, right? But this is not unusual. You guys are already getting the drift with this. Okay, so before we go down to like the microscopic level of a muscle cell and how it contracts, let's just talk about the gross anatomy of a muscle as a whole, as an organ, okay? And you guys are really gonna become familiar with this in lab when you're identifying your origin, insertion, and actions. So muscles, every muscle in the body, has what's called an origin and an insertion. If we were to define the origin, The origin of a muscle is the least movable part of that muscle. Okay, so the origin is the least movable part, which means the insertion is the most movable part, or more movable part, okay? The belly of a muscle is in between the origin and the insertion. So a muscle belly is like the meat of a muscle. Boom, smack dab in the middle. Okay? And all muscles perform specific actions. Okay? And an action is defined as a muscle's insertion moving closer to its origin. Okay? So, we'll use an example, okay? What did we call this action again?
Flexion.
Flexion of what?
The elbow.
The elbow, or you can say the
Forearm.
forearm, okay? So forearm flexion or elbow flexion. That's the action we're gonna use as an example. The prime mover is the muscle that performs a majority of that action, okay? So in this example, the prime mover is gonna be the biceps brachii. You guys are gonna learn that next week in lab, okay? Biceps brachii is the prime mover for elbow or forearm flexion, okay? What does antagonist mean? Like if you're reading a book, what's the hero, right? So the antagonist is gonna do the opposite of the prime mover. So if the biceps brachii flexes, then the opposite movement is what?
Extension.
Extension. And on the back side of the arm, we have another muscle. Anyone hazard a guess as to what that's called?
Triceps.
The triceps brachii. So for our example, if the prime mover is biceps brachii, the antagonist is going to be triceps brachii because it's on the opposite side. It does the opposite action of the prime mover. Okay. A synergist is a muscle that assists the prime mover. It doesn't do the primary action. It's not the number one mover of that action, but it will assist. Okay. So the synergist for the biceps brachii, next week in lab, you're going to identify the brachialis muscle, which is this muscle right here that they're demonstrating. Brachialis muscle, much smaller muscle, but it does contribute to elbow flexion. Okay, so it's considered a synergist. Okay, and a fixator is a muscle that stabilizes either the pectoral girdle or the pelvic girdle so that your muscles have a stable base with which to contract and perform a movement, okay? So for example, if our action is elbow flexion, all right, we need a muscle that's gonna stabilize our scapula against the thorax so that we can perform an isolated bicep curl, okay? And we're not just recruiting everything in some wild movement. It's a very controlled motion, right? So you have to have stability of the pectoral girdle in order to perform that isolated movement. So the fixator, an example of a fixator for this example would be the serratus anterior muscle, which again, we're gonna learn next week in lab. So the serratus anterior originates on the medial border of your scapula and it attaches onto the ribs of your thorax. So it keeps your scapula against the thorax, stabilizes your upper extremity, boom, now you can do your bicep curl in a controlled manner, okay? What keeps me in business is that a lot of people are very weak in their fixators. Because you spend more time doing biceps curls than you do serratus anterior exercises. How the heck do you strengthen your serratus anterior? Well, I know how, but that's because I'm a PT. And I do that all the time for my patients. Because I check their strength and they're just so weak. I'm like, no wonder you have shoulder pain. No wonder you have tendonitis. You know, it's because you're doing 100-pound bicep curls and you're not working everything else. It's like balance, right? Okay, questions about those terms? All right. Okay, so now let's dive down into a single muscle fibril, a myofibril or a muscle cell, okay? Sorry, a myofiber. I gotta make sure I'm saying this right. Myofiber or a muscle cell. So when you're looking at pictures, okay, in the next couple slides, it can get confusing as to whether you're looking at a fiber or you're looking at a filament or you're looking at a fibril, okay? If you see a cell that has a covering on it with nuclei, then you know you're looking at the level of a muscle cell, okay? If you're looking at a picture without a nucleus, without a plasma membrane, so to speak, we'll get some more terminology going here, then you're not looking at the muscle cell level. You're looking deeper to that, okay? Okay, so let's take a muscle fiber, a muscle cell. Here's a nuclei, here's the covering, okay? So I know I'm looking at a muscle cell here, and we're gonna take out a couple organelles. We're gonna take out a few myofibrils, okay? Okay. Now, we learned the plasma membrane surrounds all cells, okay? Smallest living things. Well, we're talking about a muscle cell here, and muscle cells have their own covering. That covering is called the sarcolemma, okay? So the sarcolemma is gonna be the plasma membrane of a muscle cell. Phospholipid bilayer, everything same. It is now called the sarcolemma, okay? Okay. So, next, we have what's called the sarcoplasm. What does sarcoplasm sound like? Sounds like cytoplasm, right? So the cytoplasm of a muscle cell is called the sarcoplasm. So it's the fluid inside plus the organelles, which are all the myofibrils. That's sarcoplasm. Okay. So you can define the sarcoplasm as the intercellular space. That's pretty much everything inside the cell is going to be within the sarcoplasm. Okay. Okay. Sarcoplasmic reticulum, what does that sound like? Endoplasmic reticulum, yeah. So endoplasmic reticulum has kind of a cohort in muscle cells. It's called the sarcoplasmic reticulum. So this is like the smooth endoplasmic reticulum of a muscle cell, okay? And the sarcoplasmic reticulum is super important because this is the calcium bank. Okay, this is the bank of calcium ions. And you will soon realize how important calcium is to muscle contraction. Okay? So the SR, or the sarcoplasmic reticulum, is the calcium bank of a muscle cell. Okay. All right. Now, the terminal cisterna, okay, the terminal cisterna, you can think of is the part of the sarcoplasmic reticulum is like the doors of the bank. The terminal cisterna is the doors of the sarcoplasmic reticulum where calcium is going to leave and it's going to come in. Okay, so the terminal cisterna are the doors. All right. And what's left are these T-tubules, which are these yellow structures, and they have a specific structure because they perform a specific function, which you will find soon. So they come, T-tubules come from the sarcolemma, and they invaginate and wrap themselves around the sarcoplasmic reticulum of each myofibril, okay? Now, a T-tubule and the terminal cisterna on either side of it, so that part of the SR where calcium is released, a T-tubule and two terminal cisterna is called a triad. Okay, so that's what triad means. All right. So you can see that the sarcoplasmic reticula and the terminal cisterna, the doors, basically wrap themselves around each myofibril. Because again, this is a calcium bank and calcium is needed for muscle contraction. So the SR and terminal cisterna are going to be everywhere. Okay, all along the length of a myofibril. Okay, so at the top, again, we're looking at a muscle cell. We can tell because we see nuclei and we see a sarcolemma. So that's a muscle cell. We're going to pull one organelle out, and that organelle is called a myofibril. Okay, that myofibril is made of two myofilaments, which are two proteins, actin and myosin. So you can see how each muscle cell is just stuffed with protein fibers. It's stuffed with myofibrils. There's mitochondria squeezed in between, but there's not much else. A muscle cell is just stuffed with these protein fibers, okay? And these protein fibers have a specific orientation. And it's the orientation of these muscle fibers, of these protein fibers, it's the orientation of the protein fibers that gives skeletal muscle and cardiac muscle the striated appearance. So the striped appearance that you're seeing, those are actin and myosin, okay, that interlace themselves and they create these stripes, okay? All right, so within a myofibril, okay, we're gonna look at just a piece of the tube, okay? And within the piece of the tube, we have two different protein fibers, two different myofilaments. We have actin and we have myosin, okay? If you hear the thick filament, we're talking about myosin. If you hear the thin filament, we're talking about actin, okay? And that's just based on the anatomy of the protein fiber itself and how it orients itself within a muscle cell, okay? Okay, so, yeah, what we're gonna do is we're gonna draw the fundamental unit of muscle contraction, which is called a sarcomere. And I have a slide that has that term on it, so it's coming. Okay, so what I want you guys to do, okay, is draw kind of a zigzag line, one on either side. Color is irrelevant, okay? So such that the points meet and then they go the opposite way and then they meet and then they go the opposite way, okay? Just a couple zigzags. Then what I want you to do, I used red and blue just because it's similar to the pictures that you see in your textbook. So if you have red and blue, great. If not, it's okay. Doesn't matter. Okay. The red fibers, okay, I want you to draw thick. Okay, so these are thick fibers. So let me modify them. I'm gonna make them thicker. Okay, so I'm gonna make mine thick. Thick red fibers. The red fibers are not going to touch the zigzags, but they're going to orient themselves in between. Okay. And again, we're not all artists here, it just we're getting the basic concept. All right, so we draw a couple thick filaments. From there, at each point, okay, we're going to draw a thin filament, and I use the color blue, okay? So for the points that are facing each other, you draw a thin filament on each side, but they're not touching, okay? There's a space in between. There's a space in between. And then on the outskirts, you draw one, okay? Okay. All right, so that you just drew, are called Z discs. Okay, so each squiggly line is called a Z disc. Okay, The red fibers that you drew are myosin. Okay. The blue fibers, or whatever color you use, doesn't matter, are actin. So myosin is going to be the thick filament, actin is going to be the thin filament. Okay? And the distance between Z discs is called a sarcomere. Okay? And I have this on another slide, so you don't have to write all that down, but the sarcomere is the fundamental unit of muscle contraction. This is as microscopic as we can get. Okay? You see Z discs, zigzag, z, okay? One on either side. You see a thick filament here, myosin in red. You see the thin filaments that touch the Z disc, but they don't touch each other, so there's a space in between them, okay? And the sarcomere is the length of one Z disc to another, okay? So all along the length of a myofibril, which is the organelle of a muscle cell, are millions of sarcomeres, all in parallel, okay? So this is the fundamental unit of muscle contraction, all right? Okay. Sarcomeres have the ability to shorten, okay? And if enough sarcomeres shorten in parallel, all along the length of the muscle as an organ, what happens to the muscle?
Contraction.
It shortens or contracts, right? Exactly. A repetition, which is nice. Okay, 2 protein fibers that we know are associated with the sarcomere. Thick filament is myosin, okay? Red in the top picture. Thin filament is actin, okay? Blue in this picture, okay? Now, if we look at this bottom picture here, we see that actin, okay, blue, kind of takes on the appearance of a double string mother of pearl. So say you have a pearl necklace and you got 2 of them, and you're just stringing them around each other, okay? That's the structural look of actin as a protein, okay? So in pictures, or in videos, if you watch videos, you're gonna see this double strand mother of pearl, and that's actin, okay? Myosin, okay, in pictures, all around, think of it as a 3-dimensional like tube, right? Myosin has these little arms and heads that stick off of them. Myosin all the way around the length of the protein fiber, 3-dimensionally, top, bottom, sides. You have all of these what are called myosin heads that stick out of the myosin fiber, okay? On the heads, on the myosin heads, okay, are binding sites for ATP, okay? And what is ATP? In layman's terms, drive muscle contraction. And what organelle makes ATP? Mitochondria. That's why you see mitochondria interspersed in the myofibrils within a muscle cell. So mitochondria are constantly making ATP. Constantly, okay? So ATP has its own special binding site on those myosin heads. Now, ATP itself, the energy is stored within the molecule itself. So we have to break down ATP into usable pieces, okay? The usable pieces of ATP that we need are ADP and PI, which is just inorganic phosphate. So we need ADP and an inorganic phosphate. So we need ATP to be broken down because adenosine triphosphate has the 3 phosphates attached. we need to break it down into a 2 and a 1. And that's gonna deliver and generate the energy that's needed to drive muscle contraction, okay? But in order to break ATP down into ADP and PI, we need an enzyme. That enzyme is called ATPase, okay? So ATPase is kind of floating around those myosin heads too. And he's just waiting for ATP molecules to bind to the myosin head. Okay. Now, because the myosin heads have binding sites for ATP, okay, actin then is going to have binding sites for myosin. Okay. And ideally, right, when myosin binds to actin, okay, it's going to pull actin towards the center of the sarcomere. Okay, so let's go back to our picture here. And again, color is irrelevant, so I'm just going to pick green as my example. And I'm just gonna draw some little heads coming off of myosin, heading towards actin. Okay, and they're on both sides, there are multiple, there are millions and millions, so we're just, it's going to pull actin towards the center of the sarcomere. Okay, that's why actin fibers do not touch each other, there's a space in between, okay, to allow for shortening. Okay, now a muscle can only shorten so much, right? You can do a bicep curl, but you come to an end. You can't just keep going. Like, it comes to an end, okay? Max contraction. And that's when the two pieces of actin in a sarcomere hit each other. When they hit each other, boom, you've reached maximum contraction, okay? But what does that do to the Z discs? Does that bring them closer together? Brings them closer together, right? So a sarcomere shortens when it contracts. And if enough sarcomeres in parallel contract inside of a muscle as an organ, the muscle itself will shorten, right? So it's, I mean, we're not even done with the story yet, but it's already mind-blowing. Like, wow, okay? We haven't even talked about calcium yet. Okay. Now, let's just finish labeling this picture. Okay, so the distance in between, color here, right? The distance in between the two myosin filaments is called the H band. Okay? Or you might see H zone in your references. Either one is fine. Okay? The length of a myosin filament is called the A band. Okay? And the distance between adjacent myosin filaments in different sarcomeres is called the I band. Okay. When a sarcomere shortens and those myosin heads pull actin towards the center of the sarcomere, what happens to the H band? Does it get larger, smaller, or stay the same? It gets smaller. Okay, good. Okay. When a sarcomere shortens, okay, all along the length of a muscle fiber, muscle cell, what happens to the I band? Does it get longer, shorter, or stay the same?
Longer. Shorter.
Here longer and a hair shorter. Sarcomeres all getting shorter, what does that do to the I band? What does that do to the distance between the myosin fibers? It makes it shorter, right? Because they're coming closer together. Because this Z disc is gonna come closer to the other zetus that's on the other side. So it's bringing the entire sarcomere closer to the one next door. Okay, so the I band's gonna get shorter. Okay, what happens to the A band? What happens to the length of the myosin fiber? Does it get longer, shorter, or stay the same? Stays the same, yes, and that's a key concept. So the A band does not change, it stays the same. So essentially what happens is actin is sliding across myosin, okay? And we'll get back to that concept in a moment. I've got a couple slides for that. Okay, but questions about the sarcomere, about the parts. So know the parts and know the bands and the zones and what happens to them when a sarcomere contracts or when muscle contraction occurs. Okay? So, now, we know myosin has heads, globular heads on them. Those globular heads have binding sites for actin. Those globular heads have binding sites for ATP. The myosin heads then want to attach to actin, which means actin has binding sites for myosin, okay? But on actin, okay, because we're not in a constant state of muscle contraction, there has to be some sort of control over when we contract and when we don't. And there's two proteins that I'm going to repeat myself again in the next coming slides here. Don't remember TN or TM, it doesn't matter. The two proteins that help control muscle contraction live on actin, and they're called troponin and tropomyosin. Okay. So let's look at the next slide. Okay. Troponin is globular in its anatomy. Okay. Troponin is globular, and troponin lives on its best friend, which is called tropomyosin. Okay. Tropomyosin is more like a filament. It's filamentous. It's long. Okay. So troponin doesn't attach to actin. Troponin attaches to tropomyosin. Tropomyosin attaches to actin. Okay. Okay. So what tropomyosin ultimately does is tropomyosin will block the myosin head binding sites on actin. Because as long as those myosin heads have an ATP molecule attached to it, ATPase is going to break it down into ADP and PI, generating a huge amount of energy, and that myosin head is going to want to attach to actin every single time. But since we're not in muscle contraction constantly, there has to be control. Tropomyosin controls that by blocking the myosin head binding sites on actin. Okay? Okay, we'll come back to that concept. Okay. So in this picture, the bottom here is myosin. Color is irrelevant. So we know it's myosin because it's just showing you one myosin head. But we know that myosin has billions of myosin heads coming off of it, okay? Three-dimensionally, all the way around that protein fiber, okay? Double string mother of pearl, here is actin at the top, okay? The myosin heads have a binding site for actin, okay? Once the myosin heads have a binding site for ATP, okay? Myosin heads have a binding site for ATP. Once ATP binds to a myosin head, ATPase breaks it down into ADP and PI. That generates excitation, okay? Excitement. That myosin head wants to bind to actin, okay? And if it does, if it's allowed to, okay, that connection is called a cross bridge. And I'll repeat that again, okay? And once that connection occurs, the myosin head then pulls actin towards the center of the sarcomere, And that action releases that inorganic phosphate, so that energy gets released, and that's called a power stroke. Okay? So the way I like to think of the myosin heads is it's literally like your forearm and your fist. Okay, it's your forearm and your fist. And you can flex your fist so far, okay? So ATP binds, boom. ATPase breaks it into ATP and PI, boom. You go into moving actin closer towards the center of the sarcomere, which is called a power stroke, okay? But the myosin head can only move so far. So it moves as far as it can, boom, and then it hits the end of the road, okay? So that takes us to the next two slides. I'm just gonna go over this one. The slide after this, same thing, just a different version. So you can kind of use both of them when you're understanding the concept. Okay, so we're just gonna step by step talk this over, okay? So step one, all right, we're resting. We are not contracting, we are in a state of rest, okay? So we have the myosin head that already had an ATP bound to it. ATPase broke it down into ADP and PI, okay? But is that myosin head attached to actin? No, okay, because we're relaxed. We're not contracting right now, we're relaxed. So that myosin head really wants to, I mean, it's excited, it's ready to go, yes, let's do it. Nope, not right now, because we're relaxed. Okay, so it's not attached, we're in relaxation, okay? Once, okay, tropomyosin, like I said before, tropomyosin is that protein, filamentous protein on actin that's preventing the myosin head from attaching. It's saying, nope, not right now, we are relaxed. Once tropomyosin gives the green light to the myosin head, boom, myosin head immediately attaches and forms what's called a cross bridge. So the myosin head attaching to actin, boom, that's called a cross bridge. And once that happens, boop, an inorganic phosphate is given off, and we go through a power stroke. Bam. Okay? So again, fist can only flex so far, so the myosin head, after that PI gets released, the ADP gets released later. Okay? So he's still attached, but now his ATP binding site is empty. So what happens? Well, we've got more ATP hanging out because those mitochondria are cranking it out. And that ATP molecule that's just hanging out there says, ooh, you know, it's an open room for me, or it's an empty chair. Boom, binds to the myosin head immediately. Once it does that, that binding of a new ATP molecule will release the myosin head from actin. Because again, the myosin head can only go so far. So it releases the myosin head from actin. Once that release happens, ATPase jumps in and says, okay, I'm going to break you down into ADP and PI, and the head, what we say, cocks back into a resting or a beginning position, okay? Ready to do another, what is called cross bridge cycle. So this whole thing is called a cross bridge cycle. And it happens billions of times in order for your muscle to ultimately contract as an organ, okay? Hydrolyzing ATP means you're breaking it down into ADP and PI. So ATPase hydrolyzes ATP. Okay? So again, each myosin head has a little bit of movement, and since we have billions of them, that gradually brings actin towards the center of the sarcomere. Okay? And actin fibers don't necessarily touch, they only touch and reach that end of the road if you reach maximum contraction, because you can't go any further. That's it. Actin fibers have hit each other, there's nowhere to go. Okay, so that doesn't happen all the time. So this contraction, this cross-bridge cycle, is graded depending on what you're doing, right? Are you doing a 1 rep max at the gym? Or are you lifting a gallon of milk out of the fridge, right? I mean, if you sit and think about like The specificity of muscle contraction, it's like mind blowing. It's crazy. It's awesome. Okay. And again, you don't think about this until you're forced to study this in anatomy. Okay, so similar picture here, green myosin, lots of heads on it, okay? So this is showing the head not attaching. This is a new ATP that has bound and has released the myosin head from actin, okay? Once that ATP attaches, ATPase will, boom, convert it into ADP and PI, and if tropomyosin allows it, it will attach again and go through another cross-bridge cycle. Okay, so this is just kind of a review. So, since we're not in a constant state of contraction, how do we control tropomyosin from getting in the way of the myosin head binding sites? What tells tropomyosin what to do? Well, ultimately, calcium tells it what to do. And calcium ions love troponin. Troponin has binding sites for calcium. Okay? When calcium binds to troponin in a contracting muscle, troponin tells tropomyosin, get out of the way. We have the green line. Myosin head, boom, attaches, forms a cross bridge. And then boom, we release the PI, we go through a power stroke, and here starts the cross bridge cycle. Okay? What drives that is the presence of calcium in the sarcoplasm of a muscle cell. Okay? So in a relaxed muscle, when we don't have contraction, calcium is not present in the sarcoplasm. Calcium is in the calcium bank. And what is that called again? The sarcoplasmic reticulum, right? So, no calcium, no contraction. So if you don't have adequate calcium levels in your body, you cannot move properly. That's how important calcium is in the body. That's why we have hormones, right? That monitor and are released from organs when your calcium levels in your bloodstream are either too high or too low. Detrimentally too low is the key here, okay? Okay, so this pretty much summarizes what we just talked about, okay? So troponin and tropomyosin are besties, but troponin is also besties with calcium, all right? So tropomyosin is going to block myosin head attachment in a relaxed muscle. It's not going to allow a cross bridge to form. Once calcium enters the picture, then troponin is going to tell tropomyosin, get out of the way, we have the green light. And boom, we form a cross bridge and power stroke cross bridge cycle. Okay? So you think about the magnitude, billions of sarcomeres, this has to happen all at once, all in parallel in order for your muscle as an organ to shorten. Okay? Amazing. Okay, so what's the role of calcium? So, this is why calcium is stored in the sarcoplasmic reticulum. And it's released from the terminal cisterna. So the terminal cisterna are going to determine when to open the doors and when to keep them shut. Okay? And once those doors open, calcium enters the sarcoplasm and it's immediately going to look for troponin. It's like, where's my bestie, troponin, where are you, kind of a thing. Okay? And then once that union happens, then boom, you're triggering the events of muscle contraction. Okay. So again, this is just written form of what we just discussed. Okay. All right, so driving home the fact here. So when a sarcomere shortens, okay, We know that Z disc to Z disc, the distance between Z discs gets what? Longer, shorter, stays the same? Shorter during muscle contraction, right? What about the length of the myosin fiber? Longer, shorter, stays the same? Stays the same. What about the length of an actin fiber? Longer, shorter, stays the same? Stays the same, okay? This is why it's called the sliding filament theory. The filaments, the protein fibers, are sliding along each other, okay? Which shortens the sarcomere overall as the fundamental unit of muscle contraction. But actin and myosin themselves do not shorten. They slide along each other. The myosin heads pull actin towards the center of the sarcomere, okay? Here we go. So, relaxed, contracted. And this is pretty much driving home the parts that we drew. So the Z discs come closer, okay? What happens to the I bands? Do they get longer, shorter, stay the same? Shorter, right? Those myosin fibers in adjacent sarcomeres are getting closer to each other. And then what happens to the A band? Does it stay the same?
Yes.
Yes, it does, okay? And then what happens to the H band? Does it get longer, shorter, stay the same? Shorter. And if this is considered maximum contraction, where you've got the actin fibers, boom, abutting each other, then the H band disappears and this is maximum contraction. Okay, but we're not in a state of maximum contraction all the time, thankfully. That would be exhausting. Right. Okay. Questions? I know it's a lot to swallow. So let's kind of back off a little bit and just talk a little bit more conceptually about muscles. And this we're really going to address more in lab, okay? So when we name muscles, we can name muscles based on direction, okay? Based on location, based on size. So size, for example, we have a pectoralis major and we have a pectoralis minor. Which one is bigger? Major. We have a gluteus maximus and we have a gluteus medius. Gluteus maximus is larger, okay? The number of origins of the muscle. Biceps brachii, how many do we think we have? 2. Triceps brachii, how many? 3. Quadriceps femoris, how many? 4. There you go, okay? Shape. So you can say orbicularis oris, which is more of a circular, okay? The name of the muscle could be based on the origin or the insertion, like the deltoid muscle, inserts on the deltoid tuberosity of the humerus, okay? And then the action can actually be included in the name of the muscle, adductor magnus. Oh, okay, that adducts the thigh. All right, so again, this will really be driven home in lab. Fascicles in a muscle can be arranged in different forms. So they can be parallel, they can be circular. Pennate means feather-like. So you can have multiple feathers, like on a deltoid, or you can have like half a feather, which is unipennate, or just one feather, which is bipennate. Okay. So again, just depending on where we are in the body. Unipennate are typically in the forearm. Bipennate, like quadriceps muscle, when we get to it. Multipennate, so multiple feathers, deltoid, because again, we have a lateral deltoid, we have an anterior deltoid, we have a posterior deltoid. So it kind of fans the whole top of the shoulder, and you've got multiple feathers, so to speak. Okay, so that's what pennate means. And it's just the arrangement of the fascicles. Okay, so we've talked about muscle contraction, but we're not always in a state of contraction, okay? So we know now that it's controlled by the presence or absence of calcium in the sarcoplasm, okay? But we need to take a step back and ask ourselves, what tells the terminal cisterna to open the doors? Like, when because we're not always in a state of contraction, when does the terminal cisterna know to open the floodgates and let calcium into the sarcoplasm, all right? Well, this is where we have to talk about the nervous system, because like I said before, nerves give life to muscle. Nerves are gonna generate the excitation that's going to drive muscle contraction. Without nerves, muscles just lay dormant. You can have a beautiful muscle system, but if you don't have a nervous system to go with it, it serves you no purpose. Okay, hard to imagine. Okay, so we need to kind of cover just some of the basics of like a nerve cell, and then we need to talk about the relationship between a nerve cell and a muscle cell. Okay, super important. So with structure comes function, okay? A nerve cell is called a neuron, okay? And neurons have specific anatomy associated with them, okay? On one end of the neuron, we have what are called dendrites. I think of them as, I don't know, pigtails, hairs, whatever, coming off the head. Dendrites receive information, okay? In the middle, we have a cell body. It's also called a soma. And in the cell body is the DNA, genetic material. But the cell body is also important because it makes neurotransmitters. And neurotransmitters, one specific neurotransmitter, is important in our story. Okay. Now, the axon is the part of a nerve cell where an electrical impulse is going to travel. Okay, so it's going to travel all the way from the cell body towards the opposite end of the cell. Okay? The axon is surrounded by a fatty sheath made of the protein called myelin. And we'll talk more about that in a minute. So what myelin does is it insulates the electricity and allows the electricity to stay on the axon and not be dissipated into the environment. Okay, so it keeps the electricity rolling along the axon towards the opposite end of the nerve cell. Okay, and then at the end of that cell, opposite end, we call that axon terminals. This is where we're going to find vesicles that contain neurotransmitter. Okay. This next picture is just your book's rendition of a really pretty nerve cell. Same thing, okay? The myelin sheath along the axon organizes itself in pockets, and there's a reason for this. Okay, talk about it in a minute. Okay, so the axon hillock. Let me go back to the pretty picture here. The axon hillock is the region in between the cell body and the axon of a nerve cell. The axon hillock is where a nerve impulse or where electricity is generated within a nerve cell. Okay, so it's kind of an important structure. Okay, so if we're talking about electricity, excitability, electric impulse, action potential, nerve impulse, all the same thing, okay? It's electricity, it's excitement that's going to travel down the length of an axon, starting at the axon hillock, reaching the axon terminals, okay? I'll explain the reversal of electric charge in a minute, okay? So, how do nerve cells, and our story is, how do neurons, they communicate with each other. So nerve cells will communicate with other nerve cells. But in our story, for right now, we're talking about how do nerve cells or neurons communicate with muscle cells? How do they communicate with muscle cells? Well, you have to have two things. You have to have an electrical impulse that's generated by that nerve cell. And then that impulse somehow has to be transmitted to the muscle cell to tell it what to do. And that's called synaptic transmission. Okay, so we'll define the basic parts. Okay, so when an action potential is generated at the axon hillock of a nerve cell, it's going to travel down the length of the axon towards the axon terminals. Okay, now think of these little guys as the beginnings of a muscle cell. Okay, nerve cells get really, really close to muscle cells. But do they physically touch? No, they don't. Okay, no, they don't. And this is where neurotransmitters comes into the story. But first, you gotta have an electrical impulse from the axon hillock traveling down the axon, okay? And that traveling, what happens is you get, all I want you guys to know from this, you don't have to memorize every single step in these next 2 slides. I just want you to know that the inside of an axon, okay, overall is negatively charged. Overall it's negative, okay? The inside of an axon is negative. Once that electrical impulse is generated in the axon hillock, okay, sodium is gonna rush in. Sodium is positively charged, okay? So because sodium's rushing in, that makes the inside of the axon more positive, right? So the inside of the axon wants to reestablish equilibrium. And so what happens is potassium ions that are inside the axon are going to rush out because potassium ions are positive in an effort to reduce the inside of the axon to more negative, okay? So all the way down the length of the axon, you're going to be getting an influx of sodium and an outflux of potassium. And that allows that electrical charge to travel all the way down the length of an axon. Okay, that's all I want you to know out of this, okay? Now, in the previous picture, I showed you that myelin, which is yellow in this picture, tends to form pockets around the axon, okay? And those pockets, in between the pockets, you have spaces, okay? Well, with structure comes function, okay? So all the way down the length of an axon here, you've got sodium rushing in, potassium rushing out, okay? By creating these little pockets, this allows the electrical activity, okay, the electrical impulse, to jump from one exposed area to the next, okay? So think about it, if there was, I don't know, fire alarm went off, and we gotta get out of the room, you know? You could take just a leisurely stroll to the doorway, right? Or you can take leaping bounds and reach the doorway a lot faster. So what that does is that increases exponentially the speed at which an electrical impulse can go down the length of an axon. And we call that saltatory conduction. Spanish speakers, what does saltar mean?
Jump.
To jump, right? So that's what the electrical impulse is doing. It's jumping from open exposed area to open exposed area to get that signal down to the end of the nerve cell lightning fast. Okay? Okay, so if we think of this as the nerve cell, so this is the end of the nerve cell, here's the beginning of our muscle cell. That electrical impulse is coming lightning fast down the axon. And it hits the end of the nerve cell, the axon terminals. But there's no direct connection between the nerve and the muscle. Okay, there's a space in between here. So we need to convert that electrical impulse into something that's going to trigger the muscle cell to get stimulated, to want to contract, okay? And this is where a synapse is formed, synaptic transmission. So a synapse is essentially the end of one cell, we call it the presynaptic cell, the beginning of another cell, the postsynaptic cell, and the synaptic cleft, or space in between, all of this is considered a synapse. And this is where neurotransmitters enter the story, okay? So here we go. So, this is basically showing 2 nerves communicating with each other, but we're gonna change this to our own story. So here's the axon terminal of a nerve cell. And here, postsynaptic dendrite, just change that to muscle cell or muscle sarcolemma. Okay, just change that and modify it to our story. So you've got electricity racing down the axon, jumping from node to node, okay? Reaching the axon terminals, okay? In the axon terminals of that nerve cell, we have vesicles. And those vesicles contain neurotransmitters, okay? Once that electrical impulse hits the axon terminal of the nerve cell, it's going to tell those vesicles to commit exocytosis, and those vesicles are going to release the neurotransmitter into the synaptic cleft, or the synaptic space. And those neurotransmitters are going to have receptors for them on the muscle sarcolemma. Okay. And so those neurotransmitters are going to immediately transfer across the synaptic cleft and attach to those receptors. Okay. The neurotransmitter we're talking about here is acetylcholine. Okay. And the union between a nerve cell and a muscle cell is called the neuromuscular junction. Okay. So we're taking an electrical signal, we're converting it into a chemical signal, okay, which is the neurotransmitters. The neurotransmitters are going to attach to receptors on the sarcolemma that are sensitive to them, and they are going to generate an electrical impulse on the sarcolemma. They're going to excite the plasma membrane of the muscle cell. So we're going from an electrical signal to a chemical signal back to an electrical signal. Because a nerve cell and a muscle cell come really, really close together, but do they touch? No. Okay, so there has to be a way to transmit the signal, and that's how it's done. Okay. Now, another tidbit of calcium that's important here is that when the electrical activity hits the axon terminals, that causes an influx of calcium into those axon terminals. And that's also a trigger for those vesicles to commit exocytosis and release their neurotransmitter into the synaptic cleft, being acetylcholine. Okay, so calcium is important here too. Okay, so again, just multiple pictures, which is really, really nice. And this kind of drives home the concept. Okay, you've got a nerve cell, here's some axon terminals, okay? Really, really close to the cell, but does not touch it, doesn't touch it, okay? So electrical activity comes down, tells those vesicles, commit exocytosis. You've also got calcium influx here, okay? Acetylcholine goes across the synaptic cleft, binds onto receptors that are sensitive to it, excites the sarcolemma, and it goes down both ways. It's not just one direction, it's both ways. All the way down the length of a myofiber, okay? Now, those little yellow structures that we talked about that live in between each terminal cisterna, where all three together are called a triad. Does anyone remember what this tube in the middle is called? T-tubule, right? And I told you guys that T-tubules start at the sarcolemma, and they invaginate and wrap themselves around every single myofibril. Now we know why, because the electrical signal is going to enter the T-tubules, travel down into the T-tubules, and tell the terminal cisterna, open the doors, right? And that's gonna tell calcium, oh, here we go into the sarcoplasm, okay? Think of like Best Buy on Black Friday, right? When they open those doors, it's like flood of people going in. So that's what calcium does. Once terminal cisterna opens the doors, you get mass exodus of calcium into the sarcoplasm. And what's calcium's best friend? What protein? Troponin, right? Calcium's gonna go to troponin. Muscle cell's excited, it got the green light from the nerve cell. Troponin is gonna tell tropomyosin to do what? get out of the way and reveal the myosin head binding sites on actin. Then bam, you've got a cross bridge, and here goes the cross bridge cycle, right? And if you get enough sarcomeres in parallel, shortening, overall you get contraction of a muscle as an organ. Okay. Okay. Now, NMJ just means neuromuscular junction. We already talked about that. Okay. And the neurons that we're talking about, the nerve cells, okay, are motor. Motor because we're involved with motion. So these neurons are involved in motion, movement. Okay, so they're considered motor neurons. Now, the only problem with nerve cells is if you say fiber in nervous terminology, a nerve fiber only means the axon. It doesn't mean the whole cell. So a nerve fiber is just the axon, okay? And we'll get back to that in the nervous system. Okay, motor units, next slide. Here we go. So, the definition of a motor unit is a single nerve cell and all of the muscle fibers that it innervates. Say, muscle fibers are cells, okay? So each fiber, each muscle cell only has one neuron, one nerve cell that it communicates with. But a nerve cell can communicate with multiple muscle fibers, okay? So in this picture we have a pink nerve cell and a blue nerve cell. And the pink nerve cell is communicating to three muscle fibers, and the blue nerve cell is communicating with 2 muscle fibers. Okay? So nerve cells can communicate with more than one muscle fiber, but each muscle fiber is only innervated or only responds to one nerve cell. Okay? And we use the term recruitment of motor units, okay, because for example, one motor unit, so let's do here. Okay, so one motor unit can have 150 muscle fibers associated with it, on average, okay? So this is where, if I have a patient in the clinic, and they're coming into me after an ACL repair, anterior cruciate ligament, you already know where that is, and their quadriceps muscle on the front of their thigh is super weak and swollen, and I tell them, okay, contract it, show me what you got, and they're just like, eh, and I see like, you know, chicken effort, hardly anything, then I may choose to use an external electric muscle stimulation device that's going to provide electricity and teach that muscle that it's okay to contract and recruit more motor units for increased muscle contraction. Because the body at that point is saying, no, we're shutting this off. Something happened at the knee and we're gonna protect the knee, so we're gonna shut the muscle off. So I need to tell the body, okay, the surgery's done, it's okay to contract again. So we need to practice. So usually I start using a machine on patients post-op, but gradually, you don't have to use it because their body starts to realize, okay, it's okay again, and you get a stronger and stronger and stronger contraction. So you can assist it externally, okay? All right, so this is like a huge summary slide of what we just talked about. Okay, so you can use that as like your summary. Okay, so the last thing I need to talk about, good timing, 1 minute, is muscle relaxation. So since we're not in a constant state of contraction, we have to have a couple things fall into place, all right? The first thing is we have to remove acetylcholine from the synaptic cleft. If acetylcholine is in the synaptic cleft, it is constantly going to bind to the sarcolemma and stimulate it. So if we're relaxing, acetylcholine has to get out of the synaptic cleft. And acetylcholine esterase, which is what the E is an enzyme that breaks it down and makes it inactive. So you've got that esterase in the cleft, and when it's triggered, it breaks down whatever acetylcholine's in the cleft. Okay? We've got to stop the electrical impulse from the neuron. So that axon hillock has to just take a breather. Okay, because once that electricity shoots down to the axon terminals, those vesicles are going to commit exocytosis. So we got to put a brake on the electricity, and then we also need to get rid of calcium out of the sarcoplasm. Because as long as calcium's in the sarcoplasm, it's going to bind to its best friend, which is who? Troponin, right? So because the concentration of calcium in the sarcoplasm is a heck of a lot larger than what's in the bank now, because we opened the doors and everybody flooded out, we have to use energy, which is why we need mitochondria. And so mitochondria is going to cause active transport of calcium to be pushed back into the bank. We're relaxing now. Go back in through the terminal cisterna to the sarcoplasmic reticulum and take a breather. Okay. Done.